Okay, let's talk about the neuron firing, our action potential. We're going to use the exact same framework that you have in your packet of a hot flame. So we're going to put this action potential in action. Bum, bum, bum. I know. What would it be without fun jokes like that, right? Okay, so let's get started. So when we think about any sensory action potential, we're always going to see that it starts with some sort of sensory stimuli. And we know that sensory stimuli is going to activate a particular gate, specifically modality gates. Now, when this modality gate becomes active, it's going to change the charge within the cell just slightly. It's going to make it slightly less negative. And this is called a local potential because it's just very localized to right where that modality gate is. So right in that local area, or this local potential, is a little bit less negative. So it's not positive, but it's less negative than it was before. Now, as this occurs, we know that the flame didn't just touch one modality gate. It touched a lot of modality gates on the limb where this flame is touching. So we see all of these modality gates begin to fire. So this modality gate fires, and this modality gate fires, and this modality gate fires. And all of them together add up, and their less negative charge adds up until we get activation of a voltage gate. And that activation of the voltage gate is really where we see the action potential starting. So we had enough electrical energy change to activate that voltage gate. And that voltage gate triggers the action potential. And that action potential is going to move down the dendrite, through the cell body, and up the axon until we hit the presynaptic terminal. So our dendrites go to our cell bodies, they go to our axon, and our axon ends in a presynaptic terminal. So that's the end point of the axon. At which point we see in our presynaptic terminal, there are little pods that are holding, or little areas that are holding a bunch of neurotransmitters. And that action potential is going to trigger the release of the neurotransmitter into the synaptic cleft. Now, the cleft is a space. It's a nothing. There's nothing there. The cleft is defined by its emptiness. So when we talk about a synaptic cleft, there's nothing in there but plasma floating around. And now we have a neurotransmitter floating across that cleft. And that cleft is going to provide the space between our two neurons. So when we talk about neurons talking to each other, they don't actually touch each other. They're always separated just slightly by synaptic clefts. Instead, the neurotransmitter is going to do the talking for us. So that neurotransmitter is going to float across the synaptic cleft, and it's going to look for a ligand gate on the next dendrite. That's a matching lock and key. So if this were acetylcholine, that has, and I'm not saying that that's necessarily true, but if this were acetylcholine, we would be looking for an acetylcholine locked gate to be opened by the acetylcholine that is floating across the synaptic cleft there. So we see that ligand gate is activated, and we'll see that there is a local potential when one ligand gate is activated. And then as we activate this ligand gate and this ligand gate and this ligand gate, those local potentials are going to build and build and build until we see that we have enough energy to activate a voltage gate. And the cycle starts all over at number 5. So the voltage gate creates that action potential, only now it's on the next hand cell. So we've seen next order neuron. So we've seen this propagation of the action potential all the way up the first sensory neuron. The ligand gate opened based on the neurotransmitter on the second neuron. So we call that a second order neuron. And now it's being transmitted up the second order neuron.